Glad to meet you children through these online classes. Now in the previous video we have discussed about what are microorganisms, what are different types of microorganisms, okay, what is the role played by them in our day-to-day life. In 3.1 we will clearly mention you only about different types of microorganisms. In the next lesson for 3.2 we will discuss what are the uses of microorganisms and what are the harms caused by the microorganisms. Now in the previous video I have mentioned you that there are how many types of microorganisms? 5 types of microorganisms. First one is what? Bacteria. Second one is what? Virus. Third one is what? Algae. Fourth one is what? Protozoa. And the fifth one is fungi. Okay, and the study of microorganisms is called as what? Microbiology. And who is the father of microbiology? Anthony van Leeuwenhoek. Because he laid the foundation. His discovery is a milestone in the discovery of microorganisms. So he is given a name called the father of microorganisms or the father of microbiology. In which year? 1674. When he was observing a drop of pond water through his single lens microscope, he is able to find some moving organisms in it. Who named it as what? Animalcules, later on bacteria, and later on different kinds of microorganisms were found out. Now in this video, in this class, what we are going to discuss is there are 5 types of microorganisms. In what way we can observe them? In the previous class I have finished you about pond water and rotten vegetable, or you can take a bread slice. Once again I will repeat for you, ma. Take a glass slide. Okay, collect some pond water. Just a drop of pond water is enough here. Just a drop of pond water. Cover it with cover slip and observe it under microscope. So you can find varieties of organisms. Some is looking like that, some are looking like this, some are looking like this. So there are scattering of so many organisms in this drop of water. By this we can understand that this drop of water contains so many kinds of microorganisms. These are called microorganisms because when you take that drop of water in your hand and if you observe like this, nothing can be seen. But the same drop of water, when it is placed on the slide and when it is placed under microscope, under magnifying lens, you can observe that there are varieties of organisms moving. So through this we can find that there are not only one type of organisms, there are so many types of microorganisms. Likewise, to observe fungi, what we have taken in the previous video, we have seen through a bread slice. Now we are taking a rotten vegetable, or else you can take a rotten pickle also, anything else. Some white hyphae you select. Okay, booju pattin edaina sare, just you select that and place it on the slide in the midst. Put a drop of water and cover it with cover slip. Observe it under microscope. You can observe rod-like structures like this with a knob. These are nothing but fungi. So by this we can understand that not only on air, not only on water, not only on soil, even on rotten vegetables, okay, even on rotten vegetables or else bread, rotten food material, you can find fungi. Okay, so this is all the way how you can observe fungi. There are some visible fungi. Best example for that is mushroom. When you go to rainy season. Now it is rainy season for you. In and around your houses, so many barks of trees will be present. On that, one umbrella-like structure will develop. Did you ever observe it? That is also a fungi. By this you can understand that some fungi are visible to your naked eye. So this is a good example for fungi. Okay, ma? So this is a mushroom. Mushroom is a good example for visible fungi. Now take buttermilk. Same way. Take a slide. Put a drop of buttermilk. Now we have to heat this buttermilk on a Bunsen burner or a spirit lamp. Anything else you can take. You should heat it. You should not burn it. There is a lot of difference between burning and heating. Heating and a slight వేడి చేయటం. Burning అంటే కాల్చి నిప్పు పెట్టడం. Okay, so we are going to heat it. Okay, now after heating it, cover it with crystal violet. Crystal violet. Add a drop of crystal violet and wait for a second. Wait for a couple of minutes. Okay, and later on just wash it gently. Gently. This word should be mentioned. Why am I saying gently? If you just keep forcefully under water, what will happen? What will happen? All this also will be vanished. Nothing will be there. Understand? So what you have to do is, you have to keep this slide under water with a gentle force of water. Then the stain will remove, and now you have to cover it with cover slip. You have to cover it with cover slip. Now observe it under microscope. Observe it under microscope. You can find rod-like structures like this. These are nothing but lactobacillus. Lactobacillus. So with this lactobacillus only, what will be prepared? Curd will be prepared. Now some fungi are spoiling the food. Some bacteria are preparing the food. By this you can clearly understand that these fungi are harmful for the food. These are useful for our food. Okay, Nana? So in the next class we will have this. Right? You understood, Mama? So with the curd rice only, we will end our meal. That tasty curd will be prepared with a bacteria called lactobacillus. Okay, now you want to observe algae. You want to observe what? Algae. Now when you want to observe algae, take a slide. How to observe algae, guys? You can go to a pond. You can go to a pond. Collect the green water. Green water. What do you mean by green water? Will be pink water, blue water, green water? No. Paakuru pattina water ni we call it as greenish pond water. Or else nearby your taps, nearby your hand pumps, some green colored softy structure will be settled there when there is lot of moisture. Paakuru pattindi annitam. Okay. So if that area is selected, keep it on the slide on a drop of water. Cover it with cover slip. Cover it with the cover slip. You can observe green colored structures like spirulina. Okay, spirulina, spiromera, like that. They are nothing but what? They are nothing but algae. Okay, if you take simple pond water, you can observe all kinds of microorganisms. If you take any part of the rotten food material, either vegetable, either blood size, either a pickle or anything else, you can observe fungi. If you take lactobacillus, if you want to observe lactobacillus, take a drop of buttermilk, you can observe what? Lactobacillus, which is a bacteria. And if you want to observe algae, the fourth kind of microorganism, you have to take greenish part of the water. Paakuru pattindi. That is the meaning. Okay. So that will have algae. That will have what? Algae. And now when you want to observe protozoa, this is the first one is fungi, second one is bacteria, third one is algae, and the fourth one is protozoa. Fourth one is what? Protozoa. Take a glass of pond water. Which water, ma? Pond water. Okay. In that glass of pond water, what you have to do is you have to mix it hay. Hay and fodder. Fodder means taunu. Okay. So in Telugu we call it as taunu. Okay. So that hay should be mixed in this. Now, when you take this hay in pond water for 3 to 4 days, you have to soak them. Okay? We have to soak hay in this pond water for how many days, ma? 3 to 4 days. It becomes the decoction. Okay? And this decoction of hay should be taken on the glass slide. So, all this you are doing immediately. All this you are doing immediately. Whereas, you are going to wait for 3 to 4 days. Okay? And now, take this drop of hay with pond water and now observe it under the microscope. You can observe amoeba. You can observe amoeba. So this is amoeba is a what? Protozoa. Okay, so all these protozoa, amoeba, Chlamydomonas, Paramecium, all these comes under protozoa. So if you want to observe protozoa, what you have to do is we have to mix the same in pond water, and you have to take the experimental material, that drop of pond water. Now again take a glass of water and mix soil in this. Mix what, ma? Soil. Mix what? Soil. Okay, after mixing the soil, stir it well properly. Now all the soil particles will settle down in the glass. Now all the solution which is coming up will become somewhat brownish in color. There is a lot of difference between normal water and soil water. When you take some soil and mix it with water, what it becomes? It becomes a soil solution. Now that solution should be taken. That solution should be taken on the drop of water, and now cover it with cover slip and observe it under microscope. You can observe so many organisms that are nothing but microorganisms. When you see soil, nothing will be present, but when you observe it under microscope, you can observe some organisms are present even in the soil also. There are some microarthropods. Already I have mentioned you before. That means small minute insects which are present in the soil, not visible for our naked eye, but can be seen under microscope, are called as what? Are called as microarthropods. So in this way, what is happening? In this way, what is happening? We are observing all kinds of microorganisms through pond water, fungi through rotten vegetables, bacteria through buttermilk, greenish pond water. With this we are seeing algae. Hair in pond water makes you to see protozoa, and soil solution will make you to see that so many microorganisms are present in what? Present in the soil. So air, water, soil is the habitat for what? Microorganisms. These soil microorganisms will increase the fertility of the soil. So there are some uses of microorganisms and there are some dangers of microorganisms. There are so many harms which microorganisms causes to us. So when you take TB is caused by Mycobacterium tuberculosis. In the previous video I have explained you. Okay, TB is caused by what? Mycobacterium tuberculosis. Okay, this is a dangerous disease which is caused by bacteria. See, this is also bacteria and this is also bacteria. This is causing a disease and this is helping us to make a curd. This is lactobacillus and this is TB. In your class, few students will study very well, few can't study very well. Few will come neat to the school, few cannot come neat to the school. Few will talk more, few will talk very less. Few will be very beautiful, few will be very ugly. Like that, in the kingdom of microorganisms also, some are useful, some are harmful. You may get it out now. Miss, you told bacteria, you told fungi, you told algae, you told protozoa, you told soil microorganism. Where is virus? Where is virus? Why did not I mention you about the virus in these? Because already I told you, virus is leading which life? Parasitic life. It is a connecting link between both living and non-living organisms. Outside the host, it is behaving as what? Non-living organism. Inside the host, it is what? Living organism. So, if you want to observe virus, what should you do? You should collect the blood of a human. Now, if you take, if anyone wants to observe HIV virus, you have to collect the AIDS patient's blood. Understood, ma'am? Polio is also caused by virus. It is also present in what? It will damage the motor neurons. It is called as infantile paralysis. What it is called, ma'am? Infantile paralysis. Okay, it is damaging the nervous system. Motor neurons will be damaged. So if you want to observe polio virus, you have to collect the blood of the polio patient. Like that, if you want to observe, you can't observe it in air because it behaves as a non-living organism. You can't observe it in water. You can't observe it in soil. Virus will not live in soil. Virus will not live in water. Virus will not live in any vegetable or fruit. But it will virus. Tobacco TMV virus. We call it as tobacco mosaic virus. What is HIV virus? Human immunodeficiency virus. So this tobacco mosaic virus is causing disease to tobacco. If you collect that infected leaf and you observe it under the microscope, you can observe TMV, that is tobacco mosaic virus. So now I hope you got the point that there are 5 types of microorganisms, and in the next lesson, with this we have completed this lesson. In the next lesson, we are going to discuss what are the uses of microorganisms and what is the danger, what is the harm caused to us through this microorganisms. So meet you back in the next chapter. Until that, take care.